Hello students, in today's class I will be talking about artificial hybridization. So have you ever thought like how the scientists they create new varieties, new improved varieties which has a better survival, quality, yield, disease resistance and other desirable traits. So there are different ways to do that, and one of that strategy is the artificial hybridization. So artificial hybridization, it is done manually, it is not naturally present. It is a technique which is used by the plant breeders to generate new improved varieties. So what is artificial hybridization? Artificial hybridization is a process in which the pollen is collected from a selected parent and then it is manually transferred to the stigma of another female parent. Okay, so in this way we are preventing the chances of unwanted pollination, because manually we are taking out the pollen from one parent and we are transferring it to the stigma of another parent that is also selected. So here the chances of cross pollination or any kind of random pollination by wind, insect or anything, any other pollinators, it is avoided. So in the complete process is called as artificial hybridization. So why artificial hybridization is done? Because in this way we have selected two parents which has an improved desirable traits, and in that we have collected the pollen from one, transferred to the stigma of another, fertilization will occur, a new improved variety which has a desirable traits from both the parent will come. So that is why we choose for artificial hybridization. So it is a strategy where we collect pollen from one parent and transfer it to stigma of another selected parent. Okay, here we avoid any kind of pollination that can occur randomly. Okay, so we avoid that. How we do that? So there are 3 steps that comes under artificial hybridization. The first one is called as emasculation. So what is emasculation? Emasculation is the removal of stamen before the anther take place from a bisexual flower. So in case of a bisexual, this is a bisexual flower, there is stamen and carpel both. Okay, so it's a bisexual flower. So in a bisexual flower, manually we remove the stamen before the anther dehiscence, that is the anther will split, the pollen grains are released. So before the anther matures and the pollen grain is released, the stamen is removed with the help of forceps. So manually with the help of forceps, the stamen is removed before the anther dehiscence occur. So this we do it in a bisexual flower, but in case we have a unisexual flower, unisexual female flower, emasculation step is not needed. Understood? Because there we don't have the stamen, it's a unisexual female plant, so we already only have the carpel. So there is no need for the emasculation step. So once the emasculation is done, we directly go for bagging. Means this emasculated flower, where we have removed the stamen, it is covered with the help of a bag. Generally we use the either the polythene or the butter paper bags are available. So we cover our, so with the help of a bag we cover our carpel, so that any unwanted pollen doesn't reaches to the stigma. So in bagging, basically we cover the emasculated bisexual flower. Okay, but in case we have a unisexual female flower, we don't do the emasculation, but we do the bagging, because we have to prevent the chance of any cross pollination. Okay, we have to prevent any pollen to come either through wind or insect, and it might can reach the stigma. So we have to prevent any random pollination. So bagging step is done, and after that we move to the artificial pollination. Which means that we collect the pollen from another plant. Okay, this is another plant. We'll collect the pollen from another plant and we'll transfer it to the stigma of this emasculated bisexual flower. Okay, so here we collect the pollen from one parent, transfer it to the stigma of emasculated bisexual flower. If it is a unisexual, we transfer it to that unisexual flower, and then we again do the bagging. We again cover it, called as rebagging. We again cover it, reason being, again there is a chance that any other pollen can come in contact with the stigma. So to prevent that, we do the rebagging, and this is how artificial hybridization is done. There are 3 steps. First is the emasculation, that is the removal of the stamen before the anther dehiscence take place with the help of forceps. Next is bagging, that is we cover the emasculated bisexual flower with the help of a butter paper bag to prevent any random pollination. Then we directly move to the artificial pollination, where we collect the pollen from one parent and transfer it to the stigma of this emasculated bisexual flower, and then we directly move to rebagging. We cover this plant so that again there is no chance for any random pollination, and this is how we generate a new variety which has desirable characters from both the parents. And with this I have covered artificial hybridization. In my next video I will be talking about the pollen pistil interaction, again very important topic for the NEET aspirants. So if you are somebody who is preparing for the NEET or preparing for their class 12th boards, I really want you all to subscribe to my channel, because I will be following the NCERT line by line. If you like my video, please like, share and subscribe. Thanks for watching.